looking forward to hear from them the results that they have achieved regarding air quality and health issues related to indoor and today also outdoor air quality. So I welcome you all once again, and hopefully we are going to have more and more awareness, raise more and more awareness regarding these issues. Welcome. I'm going to pass the word to Sandra that is going to present the first speaker. Sandra, please.
Hello, everyone. Welcome. Okay, our first speaker is Sónia Gonçalves Pereira, who is a medical doctor, intern of general practice, and a PhD tenured researcher, leading a multidisciplinary group focused in two main areas: microbiome studies, particularly regarding the interplay with the immune system in celiac disease, along with biophysical-social features of the disease, and microbial stewardship and infection prevention and control, including the development of fast-paced point-of-care diagnostic devices for antimicrobial susceptibility profiling and infection risk prediction tools, as well as the empowerment of healthcare workers and society toward better practices to prevent infection and antimicrobial resistance. Conducting her research through a collaborative approach with different fields of knowledge, she seeks to excel in knowledge transfer and bring science closer to society. So, without further ado, I invite Sónia to share her expertise with us. Sónia, the platform is yours.
Okay, thank you very much, everyone. Good afternoon. And today I'm going to talk a little bit about microbial air quality. So from a project I conducted, and we are still concluding it regarding the microbial air quality in healthcare facilities. So this was a project associated with my second field of research on antimicrobial resistance. And of course, everybody in our field usually tend to forget what's going on with the air of healthcare facilities. So that was the focus of the project, to look at the microbial air quality of healthcare, a hospital and a long-term care health facility. So I'm just going to give a brief introduction to the problem, to the situation, and then our preliminary results, just so that we can discuss a little bit. Before going on, I just have to say, so I'm on my lunch break, so I have to, after my presentations, if we can discuss a little bit the presentation, it's perfect, but then I will have to leave, so I won't be able to be until the end of this webinar. I'm sorry for that. So moving on. So in terms of a hospital, just so that we all remember, hospitals have different environments. So we have critical areas, and we have sensitive areas, and we have public areas. For instance, a coffee shop, even a hairdresser. We can find all this inside the hospital building. But in terms of health provision, we go from one room that you find in your left, so one room with very few, not very crowded, and maybe sometimes with only one patient in some situations, to the other aspect you see on your right, with an environment overcrowded with people, and not properly adjusted the space to each person inside that area. So looking at other healthcare facilities, so the long-term care, it's typically associated with the elderly, but also now currently here in Portugal, we can also have some people that are, for instance, that undergo a surgery in the hospital and need some transition care from the hospital to their places, and so they can also be in a long-term care facility while they require some more active healthcare provision. So, but the thing is, in a long-term care, we have a multitude of closed environments because typically the rooms are not ventilated, and so we have not overcrowded patients in a room, but the air can be saturated. So this is the conundrum we have in healthcare. From one point of view, we have this overall, this general information. So it's well, not okay, but it's advised that we should keep the windows closed to save energy, etc., and also to prevent some health situations. But the fact is that we have lots of different people in the environment, sneezing, coughing, breathing, etc. So this may have some impact on the microbial air quality. In this image you find in your left, so you can also see these ventilation systems that also can contribute, we don't know, but may contribute to impact the microbial air quality of an healthcare facility. So giving you a little bit of a microbiology perspective, this is what we call the infection chain. So, or the chain of infection, as you prefer. So, in order to an infection occur, we need to have the infectious agents, of course, but we need to have two other players, which is the susceptible host, a person that has some immune deficiency, permanent or transitional, and someone that transmits the infectious agent. And in between, we have to have two other things. So the portal of entry of the susceptible host, and also, sorry, the portal of exit from the infected host, the person that has the infectious agents. And we also have another important aspect, which is, so I've already mentioned 5 of the 6 agents you see here in the infection chain, which is the reservoir. So typically, and as you can see, so we have here a faucet. So the water, we can think very easily that it can contribute with to this problem. It can contribute to close the chain of infection, but also the air. So I didn't find any image with the air because it's not very easy to characterize the air in a picture. But so the reservoir can be lots of different things in a healthcare facility, and also around us, of course. So the air can also be a reservoir, because bacteria, viruses, fungi can rest in the air, not only when we cough, not only when we breathe or sneeze, whatever, but they can rest in the air with, for instance, with the particles that you all know that the air also carries and suffers lots of changes if the window is closed, if the ventilation is on, if too many people is inside a place. So bacteria and viruses and fungi can also float around in the air. So that's the thing. So healthcare facility environment can also be an important reservoir of pathogens. So, and that's this slide just gives what I always meant, what I previously mentioned. So apart from the great attention that is already placed in the hands hygiene and disinfection of surfaces, and the use of sterilized equipment when we have to deal with the patients and do some procedures with the patients, and when we approach a patient, etc., this reservoir can be everything, as you say, as you see in the slide. It can be everything that reaches the patient, and not only the patient, because the healthcare worker is also stays a lot of time in the building, in the healthcare facility, and also people that visit the patients they actually interact with the environment of the hospital, so they can be colonized, and if some situation occurs, can be infected with some pathogens that they can contact with during their stay in the hospital. So lots of things are already being set up in healthcare facilities, but little attention is dedicated to the air. So that's what we try to present in the project we conducted. And these are our preliminary results. So we collected samples, microbial samples from the air of a hospital and a long-term healthcare unit. And we already see here some differences in the burden, in the microbial burden of the air. So we collected and we tested bacteria and fungi. And you can see, for instance, here an ICU, an intensive care unit, it's the lowest burden, the microbial burden that we tested. And we went to a pneumology ward and a middle term. So before going to the intensive care, and when a patient is a little bit more decompensated, we have an intermediate unit. So there is a huge difference in the microbial air quality from the intensive care units and the other wards in the hospital. Maybe because, we don't know, we didn't test it, but maybe because the intensive care unit, the ventilation system has different strategies than the other parts of the hospital. And in the long-term care, we split the rooms into areas. So the patient room, where they rest, where they sleep, where they stay most number of hours of their days, and the common areas where they're not individualized in a room, but they are doing, for instance, meals and some activities that they do during the day. So we split in those two, and the common areas where we have more people, the microbial burden is higher. And since my main focus in this part is the antimicrobial resistance, so we tested some antimicrobials, and in fact, in the majority of the bacteria that we selected are susceptible to the tested antimicrobials, but one, so this is the Astronom, which is the darkest green. The number of bacteria that resist to this antimicrobial is bigger than the other one. So we have bacteria floating in the air, and some carry antimicrobial resistance. So this, again, as I said, is the preliminary results of this project we have. And from our future perspectives regarding this project in the healthcare facilities, is how to balance these different worlds inside the healthcare facilities. So the physical quality, the chemical quality, and also the microbial quality. We are going to present our results to the institutions that were involved in the project and see all together how can we balance all these worlds, the different perspectives towards a better health, yes, a better air quality inside the healthcare facility. And so that's what I briefly have to present. Of course, translating this to our project, to Breathe In project in a classroom, similar things occur. So are the windows closed or not? Are too many people or less? And I think we are going to have interesting results when we approach all these perspectives in a classroom. So thank you for your attention. I'm free to answer some questions you may have or discuss other aspects you might find interesting.
Okay, Sónia, thank you very much for your presentation. Maybe you can unshare your presentation now.
Yeah.
And we have some time for questions since Sónia has to leave. Does anyone
I'm sorry
want
for
to...
that.
No, we already know, and it was arranged like this, no problem. So does anyone has a question? No, everyone is still shy. Okay, since you're the first presenter.
Usually I am the one who's asking questions.
Okay.
So and you don't have any today?
Today I do not have any question for Sónia. Maybe I have one for Tiago.
Okay, well, I think we can say to everyone, Sónia, that we will start collecting some samples. Okay, not next week, but no, next week. Yes, next week, in classrooms in Leiria Polytechnical Institute. So let's see if the results are similar to the hospitals, or what can we find in our sampling procedure. Do you have any idea what can happen? Sónia, are you expecting something different?
Yeah, so this, in terms of numbers of bacteria and fungi, I don't know how it will be the burden. Of course, we will find bacteria and fungi, which is okay because we all live in an ecosystem with all different species, so that's okay. My main curiosity is to see how that burden will float when we have a room empty, or with students after 1 hour, 2 hours, after the entire day of classes. So I think that's my major curiosity. Certainly we will find bacteria, certainly we will find fungi. Let's see the burden, and let's see also if we can also do those experiments, if any changes occur when we open the windows, when we open the doors, and if they correlate. I think it will be interesting to see if they correlate with particles burden also, that the other systems are also collecting, and even the CO2, etc., the humidity, everything, temperature. If we can correlate the microbial burden with all those physical chemical aspects that we are also testing, I think we will have interesting results to analyze in the end. But of course, we will have bacteria and we will have fungi. If they cause any injury to the persons, it's a whole different story. And I just, maybe to conclude my presentation, the majority of bacteria, the majority of microbes that surround us, we need them. They are okay. Only a very, very small fraction of microbes are pathogenic. So, and they are only pathogenic if our immune defenses is low. So please don't be frightened with microbiology, okay?
Okay,
And
so good.
sorry to interrupt you, Sandra. Sónia, you are more than welcome to come to EPT and try and test also in some classrooms in EPT in Tomar campus and also in Abrantes if you want to. You are more than welcome to test anything you want to and continue the study here also.
Okay, thank you very much.
We can talk about that.
Yeah,
And
for
Flavio,
sure.
I think Flavio can help you also in Abrantes.
Okay, that's great. Yeah.
Okay. Thank you, Sandra.
Okay. Yes, that was my next intervention, to say that maybe the results we can use the two institutions and share knowledge, and I think the results will be very, very interesting, combining these physical, chemical, and biological results. Okay, does anyone has another question? Okay, Sónia. Thank you very much. Thank you again. So I hope in 1 year we'll be here showing those wonderful results.
Yeah, breathe in results. Okay. Thank you very much once again.
There is a question in the chat.
Here.
There is a question in the chat.
Okay.
Okay.
Oh, it's from my colleague, Jorge Guilherme. He's asking, what type of sensors are you using to monitor?
Okay, so to check the microbial air quality, we use it's a specific equipment that vacuums the air. We specify if we are going to filter 10 liters, 100 liters, 500 liters of air, considering the dimension of the room. And so that's how we do it. We filter, the equipment suctions the air, and it delivers the filtrate to a Petri dish, and then we incubate and read. It's quite easy, the evaluation for the microbial air quality aspects.
Yes.
Do you want to ask another question to Sónia? Okay, no. I think, thank you, Edna. I did not notice the chat. Thank you. Sandra.
Okay. So thank you once again. And let's hope in the future we can discuss further all these ideas, and please move on with the webinar, which will certainly be very interesting also, and see you soon. And sorry once again for not being able to be until the end. Bye-bye.
Thank you,
Okay,
Sónia.
thank you,
Bye.
Sónia.
Bye. Okay, Cristina, now it's your turn.
Okay, it's my turn to present the next speaker. He's an invited speaker. Tiago Silva was born in the 90s and is interested in climatology, namely urban climatology and bicoclimatology. He has a degree in geography and a master's degree in physical geography and spatial planning. He is currently a PhD student at the Center of Geographical Studies, which is a research and development subunit within the Institute of Geography and Spatial Planning, of the University of Lisbon. And the project is working right now is related to evaluation of thermal comfort in Lisbon. He is a student of a dear colleague of mine, Professor Marcelo Fragoso, that cannot attend today because he's giving some lectures. And Tiago is going to speak regarding a work that is already undertaken regarding North African dust intrusions and increased risk of respiratory diseases in Southern Portugal. Tiago, please.
Okay, thank
You
you.
can share your presentation.
And thank you for the invitation to take part in this webinar. So, can you see my presentation?
Yes.
Okay, good. So, as presented, today I'll be talking about North African dust intrusions in Portugal, namely in Southern Portugal. So, this all began with a project named Geo-Sahara, which was a collaboration between the University of Thrace in Morocco and the University of Lisbon, namely my institute. And it began in 2019 and it went until the end of 2021. So this was the beginning of this project. And why was it conceived? So as we know, Sahara Desert is responsible for around 50% of total dust emissions in the world, and Portugal is particularly exposed to these Sahara dust intrusions due to its geographical position. So we wanted to assess the adverse effects on human health and namely on mortality regarding these dust events in Portugal. And these problems in human health we linked to respiratory and cardiovascular diseases. So I don't know if you know, but I will give a brief introduction of dust storms. So dust storms is a phenomena which is characterized by a very strong winds mobilizing parts of the surfaces like sand, silt, or clay, and then transporting those through the atmosphere along thousands or hundreds of thousands of kilometers, depending on the size of the materials. So generally, dust storms are different from from sandstorms, which we normally see in the movies. So dust storms have silt or clay-sized materials. They are very, very small. I will talk about them more in front. So, and dust storms have three stages: the emission of the materials to the atmosphere, their transportation, and the deposition. So there are more steps within. So let's begin with the emission. But first, let me talk about how these materials are emitted to the atmosphere. So first of all, we must go back some thousands of years back the past and think of the North Africa with forests, with lakes, with bodies of water in general. So these bodies of water, along with the ages, have decreased in size or have ceased to exist. And the sediments that were in the bottom of these bodies of water stabilized. Meanwhile, with the erosion from water or the wind, these materials, which were stabilized, became unstabilized. So with the wind, they are now emitted and carried along hundreds of thousands of kilometers. So, as I told you, these areas where the materials are generally mobilized are old bodies of water, like old dry lakes, dry valleys, or saline depressions. There are also deposits regarding wind erosion, like loess deposits or dune systems. This is a picture of Morocco, one of the rivers in Morocco. As you know, Morocco is been through a very severe drought in the last years. And as you can see, the water body has decreased a lot. So it's these kinds of water bodies that I'm talking about that existed in the past and ceased to exist, and the materials that are under on the bottom of the bodies of water are now exposed to the winds to be carried away. So this is just a picture of the main dusty areas, the main deserts, either cold or arid ones. And then, okay, let's go to the transportation. There are several stages on the transportation: the saltation process, the creeping process, the sandblasting process, but the ones that we study for this event is the suspension. The smallest materials are mobilized, as I told you, and then are emitted to the atmosphere. These materials are generally around, they have less than 20 micrometers of diameter, so they are really small, and I will show you how small they are briefly. So these are the main paths of transportation of dust around the world. The ones that we are interested about, it's from North Africa. As you can see, they can go to the Americas, either South, Central Americas, North America. But they also and mostly go to Europe, and they reach Portugal in two different branches, as I will explain you, but from the west and also from the east. Regarding deposition, I guess we have all been aware of these episodes in the last couple of weeks, at least in Portugal, and today I know that in Greece the weather is very dusty. So these kinds of materials, they fall to the surface where they reach, like Portugal or Spain or Greece or Italy, due to two main things. The gravity, of course, when the big-sized materials decrease with a distance from the origin, but also with the precipitation, which force the small-sized materials to go from the atmosphere to the surface. And I believe we all have seen these kinds of pictures with our cars so dusty because of these events. So I told you before that I was about to show you the size of these materials that we are talking about. So we have a human hair, which is around 50 to 70 micrometers. And you can see how many times a PM10 and the PM2.5, which is the materials I've been talking about, fit inside a human hair diameter. So we can see them with the human eye just on the microscope. So these are the two particulate matters that we studied on this project. And we know from the literature that they cause a several range of diseases, either in the skin, in the brain, in our heart system, in our respiratory system, and even the reproductive system. These particles, as you can see, our respiratory diseases doesn't have much defenses against them. So as you can see, the PM10, they reach the upper respiratory tract, and the PM2.5, they can even reach and penetrate our lungs. So I told you how it began. I will tell you now how it went. So in the end, I was able to do my master thesis in this topic, and I even published a paper in the International Journal of Biometrology, which you can see the reference here. So, as I told you, I studied Alentejo, namely Évora, which is in the northern, more or less, central part of Alentejo. And we accessed initially our agency for the environment, And they monitor these kinds of events in Portugal, and they have an inventory that they publish each year. So from that, we saw that we had too many days to analyze. So we had to decrease our sample, and we used satellite imagery from MODIS, either true color or aerosols. We also use meteorological data from Évora, and PM10 and 2.5 also from Évora. As you can see in the map on the left, it's the blue and the blue dot, and the green dot is the hospitalizations data caused by respiratory diseases. So, as I told you, we had from our Agency for the Environment 825 days of dust intrusion between 2005 and 2015, and we decreased with those criteria to 246 days of analysis. And what we found was that in the summer, it was when the majority of the dust reached Portugal, namely in August and September, which are the months with the most dusty days in this area. We didn't find any annual trends. So as you can see, 2011 had the majority of events, but there is no trend of increasing, so which is a bit irregular. As I told you, we analyzed the PM10 and the PM2.5, and what we found of interest was that in the winter, despite having less episodes of dust intrusion days, we had the majority of the concentration of these materials. As you can see, 45% of the days in the winter surpassed the 50 micrograms per cubic meter, so which is way higher than the legal concentrations that we have from the OMS and European Union. So to assess how these the dust storms reached Portugal, we had to study the circulation patterns. So what we found, the majority, was that to mobilize the dust from the surfaces in North Africa, we need to have a low pressure there in North Africa. So meaning strong winds, sometimes even rain. And then we need either a low pressure west of Portugal or a high pressure west of Portugal as well to bring these materials, to transport these materials. So in this scenario, we have a cyclone in North Africa and southwest of Portugal, and these kinds of circulation patterns promote a direct, like a south-north transportation system. In this one, we also have a low pressure in North Africa and a very low pressure also southwest of Portugal that also permits direct dust from North Africa to the Iberian Peninsula. Now we have an anticyclone, high pressure, usually in the Azores location, but in this case it is not. But we have a low pressure down south in the Sahel region, and then this dust, which is mobilized from the Sahel, is transported through the Atlantic via south and southwest and reaching Portugal by the Atlantic Ocean. We also have this scenario, which the dust reach Portugal from Spain, from the interior of the Iberian Peninsula. So they are also, again, mobilized by low pressures in North Africa and then transported by a high pressure upper in the atmosphere that brings the dust via Spain and reaching Portugal through the interior of the Iberian Peninsula. Also, this a similar situation. And then I also vectorized each dust stormy day with the plume of the day using the satellite imagery. And as you can see, in the summer, it is when the events are bigger. So the dust plumes are wider, they reach bigger areas. They can cover all the North Africa, almost the Western North Africa and the Iberian Peninsula. Whilst in the winter, these dust plumes are not as big, but as I have shown you before, the the concentration of the PM10 it's bigger. So fewer events, smaller dust plumes, but higher concentration of pollutants in the winter. These are the examples of the aerosol imagery and the real color imagery from satellite MODISH from NASA Worldview, which I used. And then regarding the hospitalizations data, what we assessed was in these periods, in the dust storm days that reached Portugal, most people were hospitalized urgently because of pneumonia, also because of chronic obstructive pulmonary disease, asthma, and other respiratory infections. Now, we had to model and use this hospitalizations data with our environmental data, namely the mean temperature and the PM10, which were the variables which fitted more with the inventory of dust or non-dust days. So we had to split, and we did two tests with my filtered inventory and with alpha inventory with the full 825 days. So what we reached was that during these 825 days between 2005 and 2015, there was 12% more hospitalizations, urgent hospitalizations because of respiratory diseases in the dust intrusion days than the non-intrusion days. So these kind of events promote urgent hospitalizations because respiratory diseases. So in conclusion, Portugal is very exposed to these kinds of events from North Africa. As you can expect, we should have many papers and literature regarding the theme, but we don't. So it was kind of innovative, these studies in Portugal and namely in Alentejo. And these kinds of events reach mostly in the spring, but in the summer, of course, as I told you. And in the winter, we have more pollutants, which decrease the air quality, and the risk of urgent hospitalizations is 12% higher on dust intrusion days. So thank you. I'm open to questions, and thank you once again for the invitation to be here.
Thank you, Tiago. It was very interesting, your presentation. If anyone wants to put a question to Tiago, please, you can show your hand.
Maybe I'll stop sharing.
Yes, please.
So I can see you.
Yeah. There are no questions?
Oh, you had a question, right? So
Yes, of course. Of course, I have a question. I am in the age of the question still.
We all are.
Tiago, it was very interesting to see your presentation, since it is it involves several things that I like, namely the weather patterns that are most likely to bring us the dust storms. Did you in your study find somehow, or did you study the probability of a higher occurrence of these types of events in order to promote more and more these dusty storms to come to Portugal?
No, I did not study
not
the
your field,
future
yeah.
nor the predictions, although there is a very good website which does the forecast for the dust storm days with 3 to 7 days prior to the event, I believe, which is the Barcelona Supercomputing Center, which is now aggregated with the Spanish Meteorological Institute, their counterpart for ours. And they do the forecasts with some days in advance, and sometimes I'm asked about this question because of course of the health problems, and I always recommend using that website because they do the forecast for the entirety of the Iberian Peninsula, not only Spain but also Portugal and the Canary Islands, etc. So.
Is there any correlation between the pneumonia cases that you have encountered and the type of particulate, because you make the distinction between PM10 and PM 2.5. Do you have any correlation that can give you a hint on what kind of particulate were the most dominant when those cases have increased in the urgencies in the hospital?
Yes. The most dominant is the PM10 with the 10 micrometers of diameter. The 2.5 is very difficult to assess because our cars also emit those kinds of particles and our industries. So PM10 are the ones we used for the last model, as you saw, with the temperature, because they were also the most prominent. They were the most vulgar particles. The PM 2.5, they were not very significant. And regarding the correlation, all I can say is there also a statistical association, not a correlation, because that also implies a causation. We also know that correlation is not causation, but sometimes people think about it in that way. So there is a statistical association between the pneumonia and asthma hospitalizations, urgent hospitalizations, not planned ones, because there are also planned ones, and we remove that from the data. Only urgent ones. And yes, there is a statistical association between these diseases, these problems, and the occurrence of dust in those days, in the 3 days. So not only on the first day of the exposure to these dusts, but also on the next 2 to 3 days. Yes.
Okay, thank you. I think Loisos, do you want to put a question to Tiago? I saw a hand. Stavros. It was Stavros. Sorry. It was Stavros. Stavros, yes.
Hi, Tiago. Thank you for your very interesting presentation. Actually, I have 2 questions. The first one was about the remote sensing data that you used. I think you've used AOD from MODIS. Exactly.
Yes,
So
and
at what spatial analysis was that the AOD that you used?
Sorry, I didn't understand. What kind of?
The spatial analysis of the product. It was 1 degree, 0.1 degree.
Well, I don't remember actually, but I believe the aerosol imagery had some coarsity associated with a model that calculates the aerosols in the atmosphere, but the real color run was really low from that I remember. So it was very precise. And yeah, I used Terra and Aquamodish. Besides these aerosols, they used to have an analysis, a model that calculated just dust in the atmosphere, not only the aerosols, because from aerosols we can talk about also emissions from the atmosphere to like cars, industries, our cities. So yeah, they were precise and they are free, they are open source, and it's recommended to use it a very precise way. And then when I downloaded this imagery, I used GIS software to analyze the data and calculate the plumes and their sizes and etc. I hope I was able to answer your question.
Yes, thank you. And my second question is, did you consider using a dispersion model to verify the origin of the dust that you measured in your area, in your region, like high split, for example?
Yes, I have used the vector trajectories, the backward trajectories, and in fact, I did not mention, but I had in my PowerPoint slides when I filtered those 825 days to my 246 days of analysis. I also used the high split models to be assured that those air masses were coming from North Africa and not like recirculating from other areas in the atmosphere and geographical spaces. So yes, I use them.
Okay, thank you very much, and thank you for your interesting presentation once more.
And thank you for your questions.
anyone else wants to ask a question to Tiago? No questions? Well, I'm going to end this session by inviting you to participate in our next webinar. It is going to occur on May 15th, and we are going to split our team, our Portuguese team from Polytechnic Leiria and Polytechnic Tomar, since this is going to be hosted by Nicosia University Medical School, isn't it, Edna?
No, it will be by the University of Nicosia,
Okay.
not the medical school.
But you are going to welcome us, of course. So in between the webinar, we are going also to have a workshop with several sessions. You are more than welcome to register yourself. It is free. It is going to be free, and soon I will going to update the website, the Breathing's website, with more information. Please try to connect yourselves, and we will hope to see you soon all again in our webinar and maybe in our workshop. It is going to be very interesting. We have already a preliminary program, and soon I will be giving you more news regarding these issues. Once again, thank you all for participating in our third webinar. Thank you, Tiago. And Sandra is going also to thank again Sónia for your very interesting presentations. Give my warm regards to Professor Marcelo Fragoso, please, Tiago. Okay? So, bye-bye. See you soon.
Bye.
Thank you.
Thank you.
Bye.
Bye. Bye.